Hello guys, welcome back to my channel. So let's quickly analyze the 2026 Waec specimen for biology together. So let's consider the materials needed for the practicals. So the first thing we have here is hand lens. We have beaker. We have freshly prepared iodine, sodium hydroxide solution, copper 2 sulfate, white towel, Benedict solution, and filter paper. Now, if you look at these materials carefully, you would know that there must be food tests. And because we have iodine there, we will be testing for carbohydrate because we have sodium hydroxide solution and copper sulfate, which is an alternative for burette reagent. So we are going to be doing protein tests. Then the Benedict solution as well, we are going to be testing for reducing sugar. So these should already give you an idea of the kind of food test that will be done. So beginning with our first specimen, which is specimen A, granite pod containing the seed. Now let's first look at the classification. The botanical name is Arachis hypogaea. Then for the classification from domain is domain Eukaryota, kingdom Plantae, division Magnoliophyta. Remember, for plants, it's division and not phylum. Then we have class Magnoliopsida, order Fabalis, family Fabaceae, genus Arachis, and species Hypogea. Now, don't forget, I italicized it here because I typed. If you are writing it on a paper, you should underline these botanical names separately. Other information you need to know is the type of germination. They exhibit hypogeo germination. That means the cotyledon is buried underground. It is not brought above the soil. Then for the placentation, it has a marginal placentation. That means the seeds are attached to the margins of the pod. Then the lifespan, it's an annual crop. It completes its life cycle in one planting season. Now, it is also a geocarpic crop. It means that it bears pod inside the ground. It is also a dicotyledonous plant. That means it has two seed leaves. Then it also has a taproot system. Now you may be asked the functions of the pod. Firstly, the pod is there for protection. It is there for moisture regulation. That means it regulates the moisture. It reduces desiccation when the climate on the environment becomes hotter than usual. It also has the ability to conserve water. So it is also important for the seed development. Then we have the conditions for growth. For this particular specimen to grow properly, it grows well rather in a sandy or well-drained soil. Now it requires moderate rainfall and warm climates. What are the economic importance of this particular specimen? Number one, it is a source of food, oil, and protein. It improves soil fertility through nitrogen fixation. Um, this particular plant is in the legume family, so they help to fix atmospheric nitrogen. It is used as animal feed. Yes, that's very important. You use it to feed your livestock and other animals. Then, number four, it is important for the production of groundnut oil. So all these are the economic importance of specimen A in case you are asked. So this is the next specimen, specimen B, which is crushed or milled roasted groundnut seed. Now I feel like we're going to have a lot of testing to do in this year's practical. You could have food tests likely for the presence of protein, and also there could also be a test that the inference will have to be absence of a certain food class depending on what you are asked. So this particular specimen is majorly will be majorly for food test. Specimen C is beaker containing 20 ml, sorry, that's a typographical error, 20 ml of water. Now, firstly, you need to know that water is a solvent that is odorless. It's a universal solvent that is odorless, colorless, and tasteless, and it's made up of two important elements, which are oxygen and hydrogen. Now, this specimen is likely to be involved in an experiment. Yes, it's likely to be involved in an experiment. So just expect something around the food tests and and all that. Specimen D is beaker containing 20 ml of salt solution. Now, before you go into your examination lab, there must have been a constituted um, solution of hydrogen, oxygen, and sodium chloride. So it will just be there, and it is mostly going to be involved in an experiment. Now, there are some things that are supposed to be here that are not here. So I have ruled out osmosis. Also, if we had things like maybe potassium permanganate, right, I would have said, okay, we would use it for diffusion. But then just have in mind that this particular specimen is most likely going to be used in an experiment. Remember, one of the specimen is milk. So they could say you should carry out diffusion using milk and probably record your time. But also, there is no timer in list of um, the list of materials that we are supposed to provide. So just know that the content is hydrogen, oxygen, and sodium chloride, and just expect any practicals that come with it. Specimen E, sandy soil. Now, sandy soil is formed from weathering of rock. That's the first thing you should know. Secondly, it is characterized by porosity and low capillary. That means it has low ability to take up water. It is very porous. There are large holes between them since it's a sandy soil. Then third, it is one of the it is, in fact is the poorest for agricultural practice because it has low nutritional value and fertility. Now, this particular specimen may be used for an experiment, so just have that in mind. Specimen F is esophagus to end of stomach of a small mammal. Now, this particular specimen is very tricky. The only thing you need to know is the information of the esophagus and the stomach. So let's start with the esophagus. The esophagus is a muscular ring that carries out peristaltic movement. That's very important. It carries out peristaltic movement. You can go and study more on peristaltic movement. Now, what's the function of the esophagus in this case? Firstly, it connects the mouth to the stomach. So it's like a link between the mouth and the stomach. Now, what then is the stomach? The stomach is where food is temporarily stored. In case you are asked, differentiate between the esophagus and the stomach. The esophagus connects the mouth to the stomach. Meanwhile, the stomach is where food is temporarily stored. Now, the bulk of the question will come out from the stomach. So, first, you need to know that um, in the stomach, the gastric gland secretes gastric juices, which contain two major enzymes. And these enzymes are renin and pepsin. The renin will always act on milk to form a curd, and this enzyme is very much functional in neonate. That means in little babies that still feed on milk, it helps to solidify their milk and turn it into what you call a curd. So that is the function of the renin. Then the pepsin, on the other hand, breaks down protein to peptone and then polypeptides and amino acid. Then we also have hydrochloric acid. It creates an acidic environment for these enzymes that we have mentioned, the renin and the pepsin, to function properly and also kills some lethal bacteria in the stomach. So specimen G is twig or branch of plants with mistletoe, or you can get a colored picture of a parasitic plant on its host, and this is exactly what they asked. Now, you need to know that mistletoe is a parasitic plant that derives nutrient from its host. So whether it is mistletoe or any parasitic plant you can get, just know that the relationship exhibited here is parasitism. Parasitism. So the mistletoe or the parasitic plant is a parasite that lives on the host and derives important physiological needs. Now, most parasitic plants possess what we call the hosteria that pierces through the bark of the tree to derive their nutrients. So that's what you should know about specimen G. Specimen H is evaporated milk. Evaporated milk is high in protein and calcium. Then it is most likely to be used for an experiment. So based on our materials provided, this specimen is most likely to be used for an experiment. Specimen J is fresh fish. This particular specimen has been made very simple this year. Unlike when they choose a particular species of fish, no particular species of fish was chosen. So we just classify fish into domain, kingdom, and phylum here. So it is under domain Eukaryota, phylum kingdom Animalia, and phylum Chordata. But an important question you should expect from this particular specimen is the adaptive feature. You can't escape this. This one must come out. So the first adaptive feature of this specimen is that it possesses gills for gaseous exchange on water. Number two, it possesses a streamlined body that aids them to move freely or easily in water. Number three, it possesses fins for movement in water. Number four, it possesses swim bladder for the purpose of buoyancy. Number five, it possesses a lateral line that detects vibration. So this is very important. It helps them to escape from their prey when they see that maybe an attack is coming. The lateral line signal them by giving them a certain type of vibration that helps them to migrate. Also, fishes are poikilothermic in nature. That means that their body temperature varies with that of their environment. So they don't have a static body temperature like the homothermic organisms. So their body temperature can change based on their environment. Their reproduction is sexual but exhibit external fertilization. So they reproduce sexually, but their fertilization is external, and that is why um, they lay egg, and that's why they are called oviparous animals. So they are oviparous in nature because they lay eggs. Specimen K is dry maize. So the botanical name for maize is Z maize, and you can see that I have underlined it here. So this is how it should be written in case we are asked to provide the botanical name of that specimen. Then for the classification, we have domain Eukaryota, kingdom Plantae, division Magnolophyta, class Leopsida, order Poalis, family Poacia, genus Z, and species Maize. Other important things to note: morphology. The external feature of maize grain has a hard coat, which is called the testa. You can actually peel it. So that hard coat is called the testa. Then we also have it is oval or rounded in shape. Now the embryo is present at one side. So if you look at the base of the seed, you can find the embryo there. So germination of this plant is hypogeo. So it exhibits hypogeo germination. It possesses one cotyledon, right? And the leaves show parallel venation. That's an important feature of hypogeo. That's an important feature of one seed leaves. Then it has a fibrous root system. Then for the practicals that we should expect, you should expect test for starch, okay? And you may be asked to divide or cut the specimen longitudinally, and you could be asked to draw, or you could be asked to add some iodine solution if the surface on which you added the iodine solution turns blue black color, That means starch is present, because iodine solution will always turn carbohydrate to the blue black color or the violet or deep blue color. And the reagent, like I mentioned, is iodine solution. So um, that is where our materials may come handy. Specimen L is tomato paste. Now, you may be asked the morphological feature of the paste. What can you observe? Now, it is red in color. It is semi-solid or thick in consistency. It is smooth. Um, it has a characteristic tomato smell. You may also be asked why this particular specimen is processed into a paste. Number one is that it increases the shelf life. That means it increases the um, amount of days or weeks this specimen can stay before it gets bad. So that's what we mean by shelf life. It reduces spoilage, which still relates to shelf life. It facilitates storage and transportation. So when it's processed into paste, it can easily be stored and easily be transported to every part of the country and the world. Now, two methods used to preserve this specimen. Number one is canning. Number two is airtight packaging. So you know that this specimen most times it comes in tin or it comes in an airtight package. So for the food test that may be associated with this particular specimen, um, you may be asked to carry out reducing sugar tests with the Benedict solution, and the positive result of Benedict solution test will give you a brick red precipitate after heating. But then we were not asked to provide Boston Bonner and the rest, so I don't think um, there will be heating. Also, but you need to understand, you need to know that in this particular specimen, we have different kinds of vitamins. We have vitamin C, we have vitamin A. We also have other products like water and small amount of carbs. Specimen M. So specimen M is fresh tomato, which is firm, a firm fresh tomato. So let's look at the classification. Domain eukaryota, kingdom plantae, division magnolophyta. It's an angiosperm, which means it is a flowering plant. Um, class magnolophyta, magnolopsida, sorry. Then order Solanellus, family Solanaceae, genus Solanum, and species Lycoparsicum. So the botanical name for this specimen is Solanum lycoparsicum. So the observable features here are just red, fleshy and juicy. It has a smooth skin, it's rounded or slightly flattened shape, depending on the kind of tomato that was provided for you. Now it is a berry. That's the fruit type. It's a berry. Now another important information here is that it has an axile placentation. So in case you are asked to make a transverse section that you cut from left to right or right to left, the kind of placentation you will see is axile placentation. You may be asked the economic importance as well. So it's a source of income for farmers. It is also a raw material for food industries, and is a source of vitamins and minerals. Now, reproduction is by seed, obviously. Then the tomato is a dicot plant. In case you are asked, it is a dicot plant. Specimen N is rotting tomato fruit. Now, the first thing you should know here is that they may ask you to do a comparison, or they can ask you what is the observable difference between specimen N and the previous specimen, which is the firm tomato. So looking at their observable characteristics, you should be able to differentiate the observable differences and even similarities. Now let's look at the characteristics. It is soft and has mushy texture. It's unpleasant in smell. That means it has unpleasant smell and has a foul odor. It's discolored. Sometimes it can appear brown, black, or dark patches on the skin. Now, presence of mold, you can find mold or fungal growth on them, and then they are watery and always leaking fluid most of the time. Now, this is a particular question that you will not escape. I'm sure it will come out. They will ask you what caused or what causes the tomato fruit to rot. The first thing you should know is that it is caused by an attack by microorganisms such as bacteria or fungi. The moment these microorganisms attack these fruits, they begin to spoil. Number two is poor storage condition. When you don't store them properly, they get deteriorated. Number three, physical injury to the fruit. So if you have a stab to the fruit, or it is crushed or pressed, probably because of how it was transported, then it can lead to its spoilage. Then also excess moisture as well can also spoil the fruit. Specimen P is thermometer. Now, what is a thermometer? It's an instrument used to measure temperature. They would ask you to state observable features of the specimen. So you need to know that it is long, narrow glass tube. It has a graduated scale marked in degrees. Yeah, so you can see the scale there. It has a bulb at the end. Yeah, at one end. Then it contains mercury or colored alcohol, depending on the kind of um, thermometer you were provided with. Then it also has a transparent body. So just write the what you can observe about that particular specimen. So they may ask you to state the function of the specimen. Obviously, it is used to measure temperature, and they can say you should mention places where the thermometer can be used. It can be used in the hospitals and clinic, it can be used in laboratories, it can be used in the home, it can be used in weather station. And also you need to note that this specimen could be involved in reducing sugar experiments. Yes, because when you carry out reducing sugar, you need to um insert the thermometer into the beaker while the water is heating. But then it still brings us back to the fact that we were not provided, we were not asked to provide a Bunsen burner for any reducing sugar experiment. But let's see how it goes. Specimen Q is meter roll. Meter roll. So what is a meter roll? It is a measurement instrument used to measure length or distance. What are the observable features in case you are asked to mention them? It's long and straight. It's usually made of wood, plastic, or metal. So depending on what you can see on your table to you describe. It's graduated, it has graduated marking along the length and is divided into centimeters and millimeters. And it's about a length of 1 meter, which is 100 centimeters. You can also be asked to state the uses of meter rule as it relates to biological practicals. Number one, it can be used to measure the height of plants. It can be used to measure leaf, stem, roots, or specimens. It can also be used to measure distance during field trip like ecological studies. Finally, to the last specimen, specimen R. It is the quadrat. So what is a quadrat? You need to know what the quadrat is. A quadrat is a square or a rectangular frame that is used in ecological studies to estimate the abundance and distribution of organisms in a habitat. So that is what a quadrat is. You may be asked to state this function of that particular specimen. Number one, it is used to sample or estimate the population of plants or slow moving organism in a given area, like earthworms, ants, and so on. Now, they can ask you to state the uses of a quadrant. First, you know that it is used to estimate the population size or density in a particular area. It is also used to study the distribution of organism and to compare vegetations in different habitats. And you can also be asked, how is a quadrat used in an ecological sampling? First thing you need to know is that you place the quadrat randomly on the study area. So most people will say you throw the quadrat, some will say you place the quadrat randomly in the study area. Then you count the number of organisms within that quadrant. Then you record your observation. You repeat this for several times. Then you do your average calculation. You get, so if you have 1,000 square meter of land and you throw your quadrat and it lands on a particular spot, you harvest whatever you are looking for in the circumference of the quadrat. After that, you would, from that particular value, you can now estimate the number of organisms that you can find in the 1,000 square meter of land. So good luck in your exams, and don't forget to share this video and subscribe to my YouTube channel. I'll be dropping more of your exam guide as we approach the exam dates for biology.